Assalamualaikum, welcome back to my video. So we move to the lesson learning outcome number 3, which is the mitosis. So in this video we will talking about the mitosis. So mitosis can be defined as a cell division that producing two new daughter cells, and then the daughter cells are genetically identical to the parent cell, and then the chromosome number of daughter cells are same like parent cell. There are few importance of mitosis. Number one is mitosis increase the number of cell for growth, and then number two, it is used for repairing and replacing the damaged tissue. Then number three, mitosis is to produce daughter cells which are genetically identical to the parent cell, and then mitosis is the basic for asexual reproduction of unicellular organism like amoeba. So mitosis is the part of the cell cycle in which the nucleus of the cell will divide into two. So before we proceed any further, make sure you already watch the previous video of the phases of the cell cycle. So as you know, that cell cycle divided into two major phases, the interphase and the mitotic phase. And then interphase is the time where cells grow and preparing for cell division. And then during M phase, the cell is actually divided into two. So mitotic phase is further divided into two phases: mitosis, aka the karyokinesis, and the cytokinesis. So mitosis, aka the karyokinesis, is the division of the nucleus. It is the division of genetic material, the DNA in the nucleus. It consists of four major subphases: prophase, metaphase, anaphase, and telophase. While cytokinesis is the division of cytoplasm. So in plant cell, there is a formation of cell plate because a plant cell contain cell wall, and then in animal cell, there is a formation of cleavage furrow. Like this diagram is the animal cell. So in this video, we will focusing these three important event or factor in mitosis. Alright, so number one is the important events before nucleus splitting, which is the interphase. And then number two is the organelles or structures needed for mitosis. And then number three is the steps, detailed steps of mitosis. But first, let's see the overview of mitosis. So number one here, eukaryotic cell has a multiple chromosome. So one of it represented here in this diagram. So before duplication, before S phase, the synthesis phase, each chromosome has only single chromosome. And once duplicated, once it's entered the interphase, which is containing the synthesis phase, a chromosome consists of two sister chromatids connected at the centromere, and then each chromatid contains a copy of a DNA molecule. And then during mitotic phase, during mitosis and during cytokinesis, there will be separation of the sister chromatid. The centromere will break up and forming two new daughter cells like this. Okay, so now let's talk about the important events before nucleus splitting, which is the interphase. So interphase is the time where the cell prepare for mitosis, and it consists of three subphases: G1, S, and G2. So in the nucleus, look at the diagram, there is a tiny thread, we call it as a chromatid, which start to replicate themselves in the S phase. The name of the process is the DNA replication. You see that there is a nucleolus here, and the nuclear envelope still intact. So, but the chromosome, duplicating of the chromosome cannot be seen individually because they have not yet condensed. Condensed means thickening or shorter form. And then during interphase, the two centrosome form by duplicating during the S phase, and then each centrosome contain two centrioles. Alright, so now the cells are ready for nuclear division. Okay, so that's the event before the nucleus division. Okay, number two, let's talk about the importance of organelles or structure that required for mitosis. Number one is the centrosome. So centrosome is a microtubule organizing center that assembly of the spindle microtubule. Okay, so centrosome contain two centrioles. Look at this diagram. So here is the two centrioles, and then the extending part from the centriole is the microtubule. So let's simplify it like this. So this one is the two centrioles. Okay, next is the mitotic spindle, aka microtubule. Sometimes we call it as a microtubule. So microtubule controls the movement of chromosome during mitosis. So the structure of microtubule is a hollow root. It contains two types of protein subunits, alpha tubulin and beta tubulin. So these two tubulin will roll into tube, And the advantage of this this tubulin, it can elongate by aiding the tubulin units at one end, and then they maybe can disassemble, or the term depolymerize, and reassemble back again elsewhere in the cell. And then the third one is the aster. So aster is the radial array of short microtubule that extend from centrosome. So that the one that extend from the centrosome is the aster, the star-shaped microtubule. Okay, and then number 4 is the centromere. So centromere is the specialized region of the chromosome where two sister chromatids are most closely attached. So look at this diagram. The chromosome is actually, okay, one chromosome consists of two sister chromatids. It has 4 arms, referring to the two sister chromatids. It connected by the centromere. So during a cell division, the centromere will break apart and separate the two sister chromatid into 2 new daughter cells. Okay, and then number 5 is the kinetochore. So kinetochore is the proteins that attach to the centromere that links each sister chromatid to the mitotic spindle. So during cell division, the mitotic spindle, aka the microtubule, did not attach directly to the centromere, but it attached to the kinetochore protein, okay, on the chromosome. Alright. So during cell division, the mitotic spindle attached to the centrosome at one side, and on the other side, the mitotic spindle attached to the kinetochore of the centromere on the chromosome. So in order to break the chromosome equally, so 2 mitotic spindle from opposite ends of the cell attached to one chromosome. Okay, so here is the 2 centrosome at opposite ends, because centrosome are duplicating themselves during S phase. So each mitotic spindle attached at one chromosome at the kinetochore protein of the chromosome, and during cell division, the mitotic spindle will pull the chromosome in opposite direction and produce 2 new daughter cells. So here is the diagram, schematic diagram of the all organelles or structure needed or required during mitosis. So here is the one chromosome containing 2 sister chromatids, and then this one is the centrosome, each at the opposite sides. So one centrosome consists of 2 central, and then the extending array of from centrosome is the aster, and then the microtubule are attached to the kinetochore protein of the chromosome, each of the side. Alright, this one is the non-kinetochore microtubule because it doesn't attach to the kinetochore of the chromosome. So this one is the overlapping non-kinetochore microtubule.